Hello, everyone, and welcome to the microcosm of Panther Trails CDD, presented to you by Chris Lehman. Microcosm Overview. I chose to undertake this microcosm assignment primarily to better understand the ecology of my community development district, in which I call home. What I hope to learn is to develop a better understanding of the physical environment and what impacts human activities have on such ecosystems. Microcosms can be used to help simulate natural ecosystems in controlled conditions to understand the interactions between organisms and environmental changes. Here's the nitty gritty need to know. The collection site, Panther Trails CDD. Spirogyra is the algae species. Phytoplankton and filamentous algae are different. Phytoplankton can cause algae blooms with nutrient enrichment and ideal environment conditions, such as temperature and light. Filamentous algae also causes blooms with nutrient enrichments and favorable conditions such as light and temperature. Panther Trails CDD uses a lot of fertilizers and runoff adds nutrients to the pond. Microcosm temperature has been stable at 80 degrees Fahrenheit. Phytoplankton and algae blooms can be toxic to aquatic organisms. Here's the hypothesis for my microcosm. Green algae and phytoplankton population will experience exponential growth, plateau, and double in size while taking over as the primary producer. The methods and materials that I used to create this microcosm are an old, large mason jar that held full-size pickles, and the dimensions are 6 inches by 6 inches by 11.25 inches, and a volume of 128 ounces. The length of the project ran for 8 consecutive weeks, receiving 4 hours of direct sunlight and 4 hours of indirect sunlight. It maintained a stable temperature of 80 to 85 degrees Fahrenheit. Observations and data. Over the course of this project, I have observed phytoplankton and algae experience exponential growth early and then plateau. Phytoplankton and algae took over as the only primary producer in the microcosm. The turbidity has continued at a stable rate as phytoplankton bloomed. Heterotrophs started to decline as phytoplankton and algae started blooming rapidly. Observations and data continued. The turbidity was monitored using a Secchi disc using a non-traditional method. The Secchi disc was placed behind the jar at peak direct sunlight hours and visually observed through the water from the opposite side. Here's a picture after week 4 where the turbidity seems to have plateaued, and we can barely see the outline of the Secchi disk circled above. Observations and data continued. Here I've created a simple turbidity graph, where the y-axis represents turbidity in percentage from 0 to 100, and the x-axis represents time over the course of 8 weeks. Each plot point correlates with a Wednesday of each week. During the first 4 weeks, turbidity increased rapidly as phytoplankton and algae bloomed. After week 4, no change was observed and indicated turbidity plateaued. Observations and data continued. Here I've created a simple multi-data line graph to show the observation of heterotrophs expiring when compared to phytoplankton and algae growth. The y-axis represents the growth and decay from 0 to 100. The x-axis represents time over the course of 8 weeks. Each plot point represents a Wednesday of each week. The observation: for each subsequent week that phytoplankton and algae bloomed, the heterotrophs declined, until the growth of the phytoplankton and algae finally reached a plateau. Basic water quality data. It's important to note 2 additional samples were taken from Panther Trails Pond at the time of microcosm construction to draw basic water quality parameters. pH was measured at 8 parts per million. Alkalinity was recorded as calcium carbonate and was low at only 25 parts per million. Alkalinity acts as a buffer to resist rapid pH changes. Nitrite was recorded as less than 0.05 parts per million. A low nitrite level would indicate the nitrogen cycle is working well in this system. Ammonia was recorded as 0.05 parts per million. This is good for a pond, but closer to 0 parts per million is best. Ammonia toxicity rise the higher the pH is. These basic water quality parameters indicate that Panther Trail's CDD pond is a healthy, functioning ecosystem with a well-balanced nitrogen cycle. Major findings. The major findings in the project was, if given favorable conditions, such as adequate nutrients, high temperatures over 80 degrees Fahrenheit, and adequate sunlight, could result in significant and exponential growth of phytoplankton and algae. Unchecked, algae growth could result in toxicity, nutrient depletion, and oxygen depletion, creating a habitat difficult for other organisms to thrive in. Observations were that as algae and phytoplankton experienced rapid growth, heterotroph mortality rose, such as the apple and mystery snails, as well as mosquito larvae. Major findings continued. During the microcosm experiment, the phytoplankton and algae began to experience exponential growth, taking over as the primary producer, causing turbidity to increase, as well as creating a thick mat of algae that blanketed the entire surface. This resulted in the expiration of several heterotrophs and the unknown aquatic plant. Temperatures remained stable and above 80 degrees. Hypothesis support. So not only did the observations support my hypothesis, but it also went beyond what my initial hypothesis was. The phytoplankton and algae did in fact experience a rapid exponential growth phase and then plateau, as well as taking over as the primary producer in the system. Furthermore, there were both autotroph and heterotroph mortalities as algae and phytoplankton grew rapidly and took over the system. Conclusions and three key concepts. One key concept is species abundance, which is defined as the total number of individuals of a species within a given area. The relevance is that the Panther Trail's microcosm had a specific area that had more than one species living within. Over time, algae and phytoplankton began succession and gradually replaced the other communities as the primary producer. This led to an overwhelmingly abundance of algae and phytoplankton within the given area when compared to other species. Three key concepts continued. A second key concept is population dynamics, which is defined as changes in population size and density over time. The relevance is that the Panther Trail's microcosm started with one small colony of algae, and over time the colony grew rapidly and outcompeted other species within the microcosm for resources, such as space and nutrients. Births and deaths determine the population growth through variables such as environmental conditions, resource availability, and competition between species. Three key concepts continued. The third and final key concept is physiological ecology, which is defined as the investigation of interrelationships between organisms and their environment and how organisms respond physiologically to environmental conditions, such as temperature, nutrients, water, and gas exchange. The relevance in the Panther Trail's microcosm is algae exhibited increased growth, and this is due to its physiological adaptation to the environment it's in. Algae obtain energy through photosynthesis and is influenced by several variables such as available light, wavelength, and duration, and they are adapted to a wide range of varying light conditions. The microcosm received sufficient photoperiods to meet optimal growing conditions. Algae thrive well in a wide range of temperature and have optimal growing temperatures. Spirogyra, an algae found along shorelines of ponds in South Florida, thrive best in 80 to 95 degrees Fahrenheit. As the microcosm temperatures remained stable at 80 to 85 degrees, it was optimal growing conditions. Conclusions are being able to understand the physiological ecology of algae will provide insights into their adaptability and hardiness against environmental changes. They can not only survive, but thrive across various habitats. Conclusions. Just how are microcosms relevant to ecology? Well, there are several reasons that make microcosms relevant to ecology, such as they simplify complex systems, allowing the manipulation of variables that are difficult to isolate in natural ecosystems, such as nutrient levels and environmental conditions. They also give us experimental replication and statistical data because of their small scale, rapid generation times, and allow more data to draw a more concrete conclusion from the results. They also allow us to explore the ecological theory with controlled conditions by creating a specific scenario where we can make theoretical predictions and then verify the validity of such theories in real-time experimentation. Further research. Further hypothesis that I would like to test is if adding an algicide is a short-term or long-term solution, and how does this exposure affect the viability of autotrophs and heterotrophs? Next time, what I could do to improve upon the algae and phytoplankton growth is I could add in excess nutrients such as nitrogen and phosphorus, thereby causing eutrophication and promoting the excess growth of algae. I could also add in more soil, reduce the amount of water, build a little of the soil above the water line, and add invertebrate land-based heterotrophs such as snails or ants. Here are a few references I compiled in order to complete my microcosm experiment.